Good afternoon, my name is Rosella Brozzi and I'm a scientific officer of the 5th co-unit of EFSA, and together with my colleague Jaime, will introduce you the guidance on the characterization and risk assessment of microorganisms using the food chain. Let's kick off by giving you some background on the exercise. As you might know, microorganisms are ubiquitous and major constituents of the food and feed chain. Many of the products that you see on the screen contain or are produced with microorganisms and are subject in the European Union to a pre-market authorization, which includes a scientific assessment conducted by EFSA. So the EFSA panels are responsible for the assessment of similar products with similar data requirements, and therefore we are of the view that a single approach should be followed to conduct the risk assessment. This is why we have prepared the draft guidance. Let's have a look at the content of the draft guidance. Here you have an outline of the presentation that reflects the content of the draft guidance. We will briefly touch upon each of these aspects of the assessment that are covered in the guidance. These are scope, taxonomic identification, antimicrobial resistance and production, toxigenicity, pathogenicity, genetic modification, presence of viable cells and DNA of the microorganism under assessment in the final product, environmental risk assessment, and impact on the gut and food and feed microbiome. In the last chapter, some examples of the outcome of the assessment are presented to allow for some predictability. And we will start by setting the scene and explaining the scope of this document, which is to establish the criteria and data requirements for the characterization of the microorganisms intentionally added to food and feed, and to some extent of the products containing and produced with these, and the impact of their use on the receiving environment. As I mentioned before, it covers microorganisms used as such and used in the production of substances of interest. In the first case, we call the microorganism active agent. In the second, production organism. It also covers other types of products such as biomasses that are produced with microorganisms but where cells have been killed but the genetic material still remains in the product. We will cover different taxonomic groups and in particular bacteria, yeast, filamentous fungi, and for the first time we have set criteria for the characterization also of microalgae and protists and viruses to respond to new needs. This draft guidance should be seen as a reference document for the risk assessment of microorganisms intentionally added to food and feed across EFSA sectors. It is meant to provide support to applicants in the preparation of the technical dossiers in the context of an authorization procedure and to EFSA panel when assessing these dossiers. To ease the reading and favor full comprehension of the document, the products that are under scope of each section are specified at the beginning of the section. The first aspect of the characterization is the taxonomic identification. The microorganisms under assessment should be in general identified at species level using internationally recognized codes of nomenclature and whole genome sequencing data analysis, except for microalgae and protists, for which a combination of morphological and DNA sequencing information of specific markers can be applied. The details on how to conduct and report the analysis for all taxonomic groups are established in the EFSA statement on the requirements for whole genome sequencing analysis that was published originally in 2021 and has been recently updated in 2024.
Now we will talk about the block of antimicrobial resistance and production that consists in three sections: antibacterial resistance, antimycotic resistance, and antimicrobial production. With respect to antibacterial resistance, the general principles stays the same as currently practiced in EFSA. The baseline is that the products intentionally added to food or to feed should not contribute to the already existing pool of antimicrobial resistance in environments. The section applies to bacteria and phages, including their host strains, and the relevant antibacterial substances are those listed in the latest compendiums of clinically relevant antimicrobials from both the World Health Organization and the World Organization of Animal Health. The hazard to be identified is the presence of acquired antimicrobial resistant genes as defined in the statement from the EFSA Biohazard Panel of 2023, which established the way to distinguish intrinsic from acquired antimicrobial resistant genes. The ways to test antibacterial resistance are a combination of genotypic and phenotypic analysis. Both types of analysis follow current practices in EFSA. With respect to the genotypic analysis, it consists of a search of the whole genome sequence of the production strains against at least two maintained database of antimicrobial resistant genes, reporting results of hits with at least 80% of identity and 70% of coverage with respect to the gene found. The phenotypic analysis consists of antimicrobial susceptibility test, in which the strain is challenged against the different antibiotics in serial dilutions until the minimum inhibitory concentration is found, and this is crossed against a set of cutoff values of MACs for different types of bacteria, which are provided in the guidance. The strain will be resistant if its MIC is higher, equal, or higher than the cutoff value of the corresponding species or group of bacteria. The way to proceed with respect to these two analyses is explained in the guidance with a workflow, which starts with always whole genome sequence analysis of the genome of the strain. And if matches for antibacterial resistant genes are found, in case of bacteriophages, this presence is already considered a risk. In case of bacterial strains, it will be needed to determine if this antimicrobial resistance match found would correspond to an intrinsic or unacquired genes according to the statement of the Biohazard Panel, and unless it can be non-equivocally determined that the gene is intrinsic, it will be treated as an acquired gene. The presence of acquired antibacterial resistant genes, in case of active agents, it will be considered automatically as a risk, and in case of production strain, it will be necessary to determine if the product contains viable cells or DNA from the strains in the product, and depending on the outcome, it will be regarded as a risk or as a no risk. With respect to antimycotic resistance, this is a novelty of this guidance. The general principle is that antimycotic resistance is not considered to be transmissible by horizontal gene transfer, and therefore there is no need for genomic analysis. And for these reasons, it's applicable only to active agents, of course of fungal origin, yeast and fungi, but not to production strains. The way to test antimycotic resistance is the same that antibacterial resistance. They are challenging experiments against a battery of antimycotics with the aim to identify the MICs and to compare them against the cutoff values provided in a table in the guidance, which is constructed according to the latest knowledge regarding resistance to different families of antimycotics or groups of antimycotics in the main species or groups of species of interest. Antimicrobial production. The assessment of antimicrobial productions is applicable to active agents, excluding, of course, bacteriophages, and also biomasses and production strains. In this case, production strains, host strains of phages are included, with the conditions that either do not qualify for QPS approach or are known to produce any relevant antimicrobials, or are included in the QPS list, but without qualification regarding antimicrobial production. The relevant substances, antimicrobial substances, are the same as before. Those listed as important antimicrobials with therapeutic use by the WHO and the WOAH. To study antimicrobial production, a combination of genomic and phenotypic analysis is required. In this case, one is not conditional to the other, so both analyses are required. The first one is the same as before, an interrogation of the whole genome sequence against at least one up-to-date database, looking for clusters of genes involved in the synthesis of antimicrobial substances. And the phenotypic text consists of challenge tests of supernatants of the strains, or in case of production strain, it can be also done with the final product, against a battery of reference strains to test whether the supernatant other substance can inhibit growth of any of those strains. And the results must be provided strain by strain. The battery consists of a minimum of 5 reference strains, which are sensitive to most antimicrobials. If the product is found to produce antimicrobials, it will be considered a risk in case of active agents. And in case of production strains, the relevant antimicrobials must be analyzed in the product in order to discard their presence, including sublethal concentration of the antimicrobial that could elicit resistance in the environmental microbiota in contact with this antimicrobial.
The following section regards the assessment of toxigenicity and pathogenicity of the microorganisms for which different requirements exist for the different taxonomic groups. Let's have a look at these requirements. For bacteria in general, the whole genome sequencing data should be interrogated for the presence of virulence markers using at least curated and up-to-date database. But there are some exceptions to the general rule, such as microorganisms included in the qualified presumption of safety list that are presumed safe and for which there are no further data required. Microorganisms included in the QPS list, but for which specifically qualifications apply, is another case. In this case, data to demonstrate compliance with these specifications are needed. An example of these are bacillus and related species for which a cytotoxicity test is required. The protocol to follow is described in the guidance. Another exception to the general rule is Enterococcus faecium, for which specific criteria for assessing the potential virulence also apply. Again, the details on how to conduct and report this analysis are detailed in the whole genome sequencing statement. The assessment of the pathogenicity and toxigenicity in the case of fungi should be based on the available body of knowledge and a whole genome sequence-based analysis using at least, again, one curated and up-to-date database. When the data indicate a potential of the strain under assessment to be pathogenic or toxigenic, further analysis might be needed. In the case of active agents, phenotypic tests should be conducted under conditions mimicking the production or use of the product. In the case of production strains and biomasses, quantitative analysis of the metabolite of concern should be conducted on the final product. In the case of viruses, the host range effectivity should be indicated as well as the absence of adverse effects for the non-intended species. For phages, host range should be determined phenotypically on a representative set of strains belonging to the target and close related bacterial species. In addition, the nature of the phages should be established via whole genome sequence-based analysis. In particular, the whole genome sequence data should be interrogated for the presence of genes coding for toxins and other virulence factors, coding for lysogeny, and also for the presence of genetic elements known to be involved in transduction. As I mentioned before, one of the novelties of the guidance is the establishment of criteria to characterize microalgae and protists. The potential pathogenicity or ability of the strain under assessment to produce harmful metabolites should be assessed based on body of knowledge. When the data indicate this potential, phenotypic test should be carried out on the cell biomass and cell culture supernatants. It should be noted, however, that depending on the output of this analysis and on the regulatory framework, further data might still be required. The fourth aspect addressed by the guidance is the assessment of the genetic modifications when applicable. The genetic modification should be fully described to allow a proper safety assessment. This should include the scope of the modifications and the description of the traits and changes in the phenotype and metabolism of the microorganism resulting from the genetic modifications. The details on how to conduct and report the analysis are also present in the EFSA statement. It is important to reiterate the concept that the QPS approach can still be applied to genetically modified strains belonging to the species included in the QPS list, provided that the genetic modification introduces no safety concerns. In some cases, depending on the nature and characteristics of the microorganism and microbial product under assessment, and on the regulatory framework, there might be the need to investigate the presence of viable cells and DNA of the strain in the final product. The guidance establishes that analysis should be conducted using culture-based methods, include the relevant controls, should guarantee the recovery of stressed cells and germination of spores, and it also establishes the amount, number, and type of samples to be tested. Similarly, and when required, the presence of DNA of the microorganisms in the final product needs to be investigated by means of a PCR, allowing the detection of at least 10 nanograms of total DNA per gram or milliliter of product. The guidance describes again the type of and amount of samples to be tested, the target gene to be amplified depending on the type of products, and the need for relevant controls and sensitivity tests.
Now we're going to talk a few words about environmental risk assessment, which is something new. The assessment of the environmental impact applies to biomasses and active agents. The environments considered in the assessment will be all exposed to the product except human and animal guts, for which there is a specific section later on. And the rules of exposure are those primary, so those directly related with the intended use of the product, and secondary, which are those indirect, for example, water streams. To assess the environmental impact, the areas of risks as defined in Annex 2, Section D1 of the Commission Directive on Environmental Risk Assessment of GMOs are considered. For biomasses, only horizontal gene transfer applies, whereas for active agents, all of them apply, which are persistent and invasiveness, horizontal gene transfer, effects on target and non-target organisms, effects on humans and animals, effects on biogeochemical processes, and effects of management techniques. The guidance establishes a series of exceptions and defines the cases in which environmental risk assessment does not mean to be done in the whole or part of it. For example, non-genetically modified plant protection products and non-GM active agents, which are QPS, as well as non-GM active agents, which are common in the reserved environments, and biomasses not containing genes of concern will not need to follow the environmental risk assessment. And for each specific area or risk, there are also cases which are exempted. For example, GM active agents which interact solely only with the target organisms do not need to be assessed for effects on non-target organisms. Now, there's a section on the impact on the gut and also food feed microbiomes, which affect active agents and biomasses for the assessment of their impact in the gut microbiome in the context on its intended use, but also applies to GM active agents and GM biomasses when they are added to food and feed for the impact in the possible effects in the microbiome already residing in that food or feed. The areas of risk considered are for biomasses again, horizontal gene transfer only, and for active agents, persistent and invasiveness, horizontal gene transfer, and effects on humans and animals. And in the same case as before, there are indications on the cases in which, depending on the circumstances, this assessment does not need to be done. For example, non-GM QPS active agents or biomasses which are obtained from QPS strains that not need to follow this section of the guidance. And there are also exceptions per specific area of risk. For example, when the genetic modification consists of an insertion conferring traits which are already present in the gut and the microbiomes resident in the food or feed, the horizontal gene transfer area of risk will not need to be considered. Finally, there is a section of outcomes, which are aimed to provide predictability as much as possible on the outcome in the risk assessment. And before the many possibilities covered by the guidance and the many possibilities that microbes can be used in food and feed, not all the cases can be covered. It's only limited to those concrete cases for which it is possible to provide predictability on the outcome of the assessment. For example, in a case of an active agent, even if it does not qualify for QPS, but if it can be shown that it's not a pathogen, does not contain antimicrobial resistant genes, does not produce harmful metabolites, does not have negative effects in the gut microbiome or in the environment, no risk is expected and the product will be considered safe. However, if any of those conditions is not met, for example, is a pathogen or has antimicrobial resistant genes or has produce any harmful metabolite or has negative effects in the gut microbiome or in the environment, the active agents will be considered to pose a risk. With respect to production strain, the risk is related with the final product and is the same when it can be produced. When some predictability can be done, the cases will be covered. For example, production strains which does not carry antimicrobial resistant genes or does not produce harmful metabolites, in this case the product will be considered safe. If the production strain carries antimicrobial resistance genes, the safety of the product will be conditioned to the presence or not of DNA of the production strain. And with this, we finalize this presentation. Thank you very much